We're going to start looking at what's important in Experience 2, and basically Experience 2 says, how do we see bacteria? We talk about staining. When we look at the size of things that we're going to deal with, you can see over here all sorts of things that go from the tiny little atom on the left-hand side all the way up to a great big tree. Depending on what it is, we need different ways of being able to observe it. This uses different types of microscopes. We're going to use a compound light microscope that really, for our point of view, goes from about a micron up. And you can see in here that we've got some photographs of some different materials that were all taken with a microscope. So we need to be able to differentiate things because of the size they're at. Why do we stain bacteria? Well, we got some reasons here. It says bacteria cells are very small, 1 to 10 microns. They are clear or transparent, as are most microbiological specimens. So we apply stain, which is really a colored dye, to improve contrast and increase visibility. That's why we apply stains, and it's a very easy way to look at it. In the atlas, it says microbes are too small to be seen by the unaided eye, and because cytoplasm is essentially transparent, viewing cells with standard light microscope is difficult without stains to provide contrast. Once stained, we can look at things like cell morphology and size and arrangement and other things like that. Now let's look at some prokaryotic shapes. There are a few basic shapes that we always see. Coccus, the plural is cocci, is a round shape. It's basically 1 to 1. They look like these little tiny spheres all next to each other. We have the cocobacillus, which is about twice as long as it is wide. We have the bacillus, the plural is bacilli, which is about 3 times or more longer than it is wide. And if you look at bacilli, they often look like these little tubular shapes. We have the vibrio, which is kind of a comma shape. Not really regular, but rather it tapers to one end. We have what is called the spirillum. The spirillum is a curved shape where it's a kind of bacillus that has been twisted or wrapped around. The spirillum often has a corkscrew-like shape and polar flagella. These are not flexible. The spirochete is elongate and thin. It's kind of twisted on an axis so that it gives it more of a stretched out elongate spring kind of look. These are very flexible. And we have pleomorphic, and pleomorphic says that the shape varies. One of the things we often talk about is cellular arrangement. In this particular case, if you look at a coccus can divide on one of 3 different planes. You call it an XYZ axis. If you get single division, we get single ones like that. You can have single ones where they kind of hold together, and we call that a diplococcus or a diplococci. You can have them where they form together in chains, and we call that a streptococcus or a streptococci. You could have them where they divide in 2 planes, but they hold together, in which case we'd call it a tetrad. Or 3 planes and hold together, and we call it a sarcina. And this is kind of a packet of 8, and you can get these in 8s and 16s going up like that. Or you could get them dividing in 3 planes where the 3 planes really aren't at right angles to each other. Then we would call that a staphylococcus. With bacilli, since they're elongate, you can get the single bacillus, you can get a diplobacillus, you can get a streptobacillus where they divide down like that. But again, you won't get much different than that because they're only dividing kind of like links on a sausage. We have a palisade, and a palisade is where they kind of go up and they stack and they kind of stay together. That's kind of difficult to see unless you're very careful when you make the preparation of these. One of the things we deal with is preparation of a bacterial smear. Now watch the video on that because it really shows you how to do it. This is going to give you an overview. It's get a microscope slide and clean it. You transfer bacteria using aseptic technique. You light the Bunsen burner, you flame the loop, you let it cool down a little bit, you remove the material out of the culture, you smear it on a drop of water on the slide, you allow it to air dry, then you heat fix it, draw it through the long way 3 times, and you're going to get your slide that you can use. You make the smear, you let it air dry, you heat fix it. And we show you all that in that video. Your slide should look something like this in the end. Simple staining, when we do simple staining, we add only 1 dye to the specimen. We flood the slide with dye. We let it sit for 30 to 60 seconds. We rinse it. We blot dry, and then you can see it. We also have differential stains, and differential stains add more than 1 color and that gives you differentiation based on some feature of the cell. This includes acid-fast stain, which will show up mycobacterium. The waxy wall on the mycobacterium really does not take up the gram stain, even though these should be gram positive. The acid-fast procedure allows them to take up stain. We have an endospore stain, which shows you the endospores on the inside, and you see these little openings in there. That is really where the stain was not taken up, and since the stain was not taken up, you get a look like this. We've got capsule stain, and on the capsule stain, it stains everything else but what you want to see. And by staining everything else, what you want to see, which is clear, is going to show up. And we've got a flagella stain, and the flagella stain basically makes the flagella stand out by adding thickness to it, and that way we could see what the flagella look like. We also do the gram stain. Gram stain, it says there are 4 major steps. We have the primary stain, which is crystal violet. We have a mordant, fixer, iodine, potassium iodide, which is a material we call Graham's iodine, and that basically is going to complex the crystal violet to keep it in place. Our decolorizing agent is 95% alcohol. The counterstain is safranin. The way it works is like this. We've got 2 different cell walls on these. Now you can see on both of these you have a cell membrane down at the bottom. Then on top of that you've got a layer of peptidoglycan. The layer of peptidoglycan in the bottom picture is thin. It's thick on the top picture. You add the crystal violet to it. The top picture gets flooded with crystal violet. It gets held in place. On the bottom picture, it's going to get a little in there. It's not going to get a whole lot because you've got another membrane on top of it. Then you add your iodine. It's going to complex it into position. You decolorize with alcohol. And when you do that, it's going to destroy the outer LPS layer on the bottom one, which is going to allow the stain to leak out. And on the top one, it still remains stained. Then you go and you add the counter stain, which is pink. And pink on purple leaves purple. Pink on clear leaves pink. So gram positive is purple. Gram negative is pink. General run through. 1 minute, 1 minute, less than 30 seconds, 1 minute. Watch the video and it shows you exactly how to do this. One of the things we often look at is on these 4 major steps, what color do things have? It says if you start out with these cells on the slide, you've got no color to it. You add the primary stain, it stains everything purple. You add the Graham's iodine, it leaves everything purple. You add the alcohol to decolorize it, and it's going to take the color out of the gram negative bacteria, but not out of the gram positive. And then when you add the counter stain, the pink on the purple is going to not change color. The pink on the clear is going to give it a pink color. If we have any problems with this, and this is one of those things that sometimes happen, we want to deal with a culture that ideally is between 24 and 48 hours. Sometimes that's a little bit difficult because sometimes the culture is a little older, and if you do that, you may not get a good result. If you heat fix it for too long, you will actually cook the bacteria, and that's going to leave them come out as a gram negative. Too much alcohol, which is very easy to do, we call that over-decolorization. Over-decolorization, it's going to leave it as a gram negative bacteria. If the reagents are too old, that can be a problem as well, as can if you don't add the iodine. So when you do this, you have to make sure that you do everything in the proper procedure and in the proper way in order to be able to get the best result out of it. So when we look at bacteria and staining bacteria, they're small, they're transparent. We have to add the stain to improve contrast so that we can visualize. Many different ways of doing this. This is in a nutshell as a couple. Watch the videos because the videos really show you how to do the procedures.